Okay, so our next speaker will be Jess Rubin, who researches myco-phytoremediation in UVM's Department of Agriculture, Landscape and Environment, and has a long-term ecological restoration study plot at Shelburne Farms. She is also founder and director of MycoEvolve, which offers an array of services including ecological inventories, permaculture consulting, habitat enhancement for pollinators, and soil fertility restoration, in addition to offering workshops and courses on these topics. The title of her talk today is Innovations in Ecological Restoration of a Degraded Riparian Forested Buffer, Investigating Myco-Phytoremediation.
So yeah, I also work at UVM in the ALE department, and just want to take a moment to acknowledge that we're on the unceded Abenaki territory, and what does that mean as practitioners and earth workers in terms of our responsibility. It's a constant living question. So we'll be talking today about these innovations in restoration of a degraded riparian forested buffer, and looking at this technology, which is based on old relationships. So today I'm going to start the timer now, moment. We'll be talking about ecological restoration limitations and possibilities. I guess I'll just do both. Socio-ecological reconciliation, we'll be talking about this case study. Non-chemical removal, we'll be talking about glyphosate, mycorrhizal natural history and ecology. Commercial versus endemic mycorrhizae, looking at some data, phosphorus, DNA, and plant diversity. Phytoremediation, the honorable harvest and cyclical coppicing, and hopefully get to research questions and time for some questions. All right, so in the field of ecological restoration, I think I'm just gonna have to look up here because it's not showing up here. In the field of ecological restoration, we have some limitations. It's impossible to restore reference communities and repair all levels of harm on the socio-ecological. So they're just starting with this fact that we've inherited these degraded landscapes, both in the ecological and sociological levels. And however, we have the possibility of reconciling that damage. So the ecotone that I'm focused on are forested riparian buffers. Particularly, we're addressing a couple of gaps. So first of all, a lot of non-native species removal is involving chemicals, particularly glyphosate. These buffers are often not able to maximize their water quality function because when they uptake that phosphorus in their plant bodies, that phosphorus goes back into the soil and water in the fall when they go senescent. And there's rarely funding for monitoring and data gathering, harvesting. Oftentimes, as we heard this morning, practitioners aren't able to think about pollinator habitat because they're just battling potentially deer, so they're limited to a quiver of trees and shrubs, and often not able to access diverse palettes as well as tracking succession. And especially in the political climate, locally and nationally, rarely are people thinking about the original peoples of this land and how to provide them access and harness their wisdom and bring them in collaboration. So we attempt to address these gaps through a holistic approach to address socio-environmental problems through these multifunctional riparian forested buffers. And going to this concept of ecological reconciliation, it's helpful to stop to look at this pre-colonial map of what this area looked like. Vermont Pollen Corps studies indicate that from the last glaciation succession, terrestrial succession from fungi to primitive plants to soft to hardwood trees, there were robust fungal networks. We'll talk more about these mycorrhizal networks. They were critical to nutrient exchange networks, and the way the indigenous people tended the land, which many people in the regenerative and agroforestry community adapt and follow today, tended these networks. Fast forward to the colonial era, we now have states and countries, and this is the area that I'll be talking about today in the 1840s. And one of the first huge devastations is this 90% death rate. This is not some small detail to overlook, and I feel like considering it all the time, thinking about not only the sociological devastation, but the wisdom and land practices of these people replaced by colonizer land practices. And one of the first things they did was cut down the trees. The fungi networks I'm talking about are what's called obligate hosts. They partner with these trees and shrubs. So when these trees were killed and cut, all these underground networks were killed. So it was a genocide above and below ground, which has serious implications, as we will probably already aware of. Then fast forward, we have now 1.7 million sheep in Vermont. That's a lot of phosphorus entering the soil. With railroads and refrigeration, we have increase of flock and herd size, more phosphorus going into that soil and fragmentation of the landscape through the roads and ditches. And then, like many farms, this farm accepted subsidies from the USDA government coming out of World War II technology, applying superphosphate fertilizer. So now we have what's called legacy phosphorus. And the remaining networks that were connected to the elm trees were killed when this disease came through and non-native species came through. So I would like to talk about non-chemical removal, which I understand is a hot topic in this community. And the strategy I learned was from Mike Bald of Got Weeds, and we've applied it on many landscapes in the last decade, and it works hands down all the time. 3 times in 2 seasons, you cut back that first growth, like here we did, and we left the native species in place, strip back that regrowth 2 more times, and you have a 90% death rate, and you're leaving these stumps for fungi and overwintering invertebrates and early migrants. Now, we did actually remove the stumps here and had many wonderful work parties doing that. What I'd like to just point out here is that it might not be necessary to remove the stumps. You could just do the strip back, and you can see that the plants did a great job growing around it. However, those of you who are managing 4 to 40 acres and are like, there's no way we're gonna have wonderful work parties with 100 people and good snacks, you could rent an excavator, which is something that I did just for 3 of the plots to get the metrics, and it's about 1/3 of the time. It's less than half of the fun. Still had to decompact the soil and get the Covington clay out, but it is possible. No need for glyphosate. It's a complicated chemical compound. It basically increases phosphorus cycle in soil and water and contributing complex compounds. And when these microbes are breaking it down, basically that phosphorus is now taking up place in the soil matrix, so that the phosphorus that would have gotten uptaken from the ecosystem is now going into the soil and the water. Mycorrhizae have been on the planet for 407 million years. They came around during the Devonian era, which is when plants were moving from water to land. And we know this from fossils that were found in plant roots, these organelles called arbuscles. This is a time when fungi had already been on the planet 500 to 900 million years, and so their mining abilities would make sense, would help plants obtain formerly inaccessible nutrients in very nutrient-poor, thin soil. So this relationship has been around for quite a while. And this is an arbuscle from one of our microscopy studies and vesicles. And when we first learned about mycorrhiza's ability to mine phosphorus, we were wondering, why is no one applying this to water quality? So we put together this literature review in 2021, and it highlights field and case studies where, yes, indeed, it does seem to decrease phosphorus concentrations entering water. However, the key to that is phytoremediation. Phytoremediation is a form of cleaning up toxins with plants. We know phosphorus is a macronutrient for growth. It's not necessarily a toxin, but it can be a toxin when in excess in water bodies. The key form of phytoremediation that we practice is called phytoextraction, which is harvesting those plants when they have the maximum phosphorus in their above ground body, which we found through multiple multiplicosm studies is in early August, which happens to be when the elderberry are ripe. It's a good enough time to coppice many woody shrubs, and they have enough time to harden off before the frost. If you're not familiar with the symbiotic relationship between plants and the fungi, basically the photosynthetic metabolism of plants is bringing those sugars down, feeding the roots, and meanwhile the roots, the rhizosphere, is expanding and mining formerly inaccessible nutrients, feeding that back to the plants. It's worth noting this relationship is not always mutually symbiotic. It can be parasitic. And this is based on a suite of abiotic and biotic factors, such as chemical stoichiometry and pH. One example, look at B over here, is you can see that where there's basically excess phosphorus in the soil, what happens is the plants can just get that phosphorus through diffusion, but meanwhile, the mycorrhizae can just take that carbon from the plants. And what that looks like is decreased biomass, and we've seen that in our pilot project, though we also have 2 other hypotheses why that may be happening. But again, there's a spectrum, and Nancy Johnson's done a lot of research on that. So mycorrhizae are not always the right tool for the job. When do you want to use mycorrhizae? Well, certainly in areas where you're not going to have further soil disturbance, thinner fertilizer, chemicals. You might want to plant cover crops, diverse agroforestry crops, native polycultures, orchards, riparian forested buffers. This is from a manual we published, it's free on the SARE website, of comparing when do you use endemic versus commercial mycorrhizae. And you can see that there's a lot more benefits to the endemic native mycorrhizae than the commercial. The skinny of it is this. We did DNA samples on the commercial years ago and found that what's labeled on the bag is not in the bag. Also, what's in the bag are often generalists. The problem with just generalists is they only partner with a few plants and only promote primary succession. And thirdly, oftentimes what's in those bags is not what's in your soil, and the soil is a very complex, just like we do here, of a social structure in that soil of fungi and microbes. And so you're basically introducing a whole nother community, and that can have parasitic effects. Endemic is relatively cheap, relatively easy, it doesn't take a lot of time. You do need a couple of tools and access. However, make sure you do microbial assays so you're not bringing in pathogens. Now, we've since learned some things from our DNA data that there are some amendments we need to make to this guide, and hopefully I'll have time to share those with you. On farms, you may remember this beloved ancestor of the regenerative farm community, now an ancestor to us. This was one of his last gifts to our community, this book, and in it I'm going to talk about 3 of his points about mycorrhiza's ability to increase phosphorus acquisition in plants, plant community composition, and ecosystem succession. I'm going to use this Shelburne Farms case study to talk about these topics, and I want to just emphasize that the 4 objectives of this project are to increase phosphorus mitigation, to enhance pollinator habitat, to facilitate rematriation, which is land back to the indigenous people, particularly the women who are the medicine keepers, at least in Abenaki territory from my mentors, and eco-literacy, because the next generation, right, that's where it's at. This case study involves a random block design on this riparian buffer that goes into Orchard Cove. Water quality samples at this area going into the lake are 18 times what Vermont water quality standards say, and phosphorus concentrations any water entering the lake should be. This is a combination of causes because of this field that's laden with legacy phosphorus from the historical trajectory I already showed, as well as this compost pile on a hill. Those of you who remember Greek philosophy, this is a Sisyphean myth. I'm basically pushing the rock up the hill. And that's because this compost pile is not getting removed, despite us creating a vegetative treatment plan, agroforestry plan. It was approved by UVM Extension, submitted to the farm, and they're not able to do it because of their complex economic infrastructure details. So we did the next best thing you do, which is a model project. We basically picked a model area that's intercepting what's called the critical source area in the lake. These are random block designs, and basically the black plots, which you'll see yellow in the data, are the Rhamnus cathartica monocultures of poison ivy, honeysuckle, and a couple other plants. That's degraded from that colonial history I told you about. The green and red plots are the plots that we restored. The green are the ones that are inoculated with mycorrhizae fungi. The red were restored the same way without added mycorrhizae. The difference between the pilot and the expanded project is that in the pilot we used commercial mycorrhizae, rookie mistake, and the expanded project was using endemic mycorrhizae, and we made many rookie mistakes in that too, but we did learn some really neat things that I hope can benefit this community. Oh, that is a view of one of the restored plots looking up here to the compost pile. So this is a little view of the plant palette, and you may have heard me use that fancy term multi-synusial this morning. It's something I learned from Tom Wessels. It's trees, shrubs, perennials, and ground cover. You can see the blooming times, March to November, the pollinator hosts, the type of mycorrhizae, and most importantly, the edible, medicinal, utilitarian qualities of those plants, because the Abenaki are the ones who are doing the harvesting, the phytoremediation. And unlike Superfund sites where plants are uptaking heavy metals, phosphorus can go in our body, it's okay, it's fine. So I just want to talk about how we gathered the data briefly so that when you're looking at the data you understand. In terms of phosphorus metrics, using an Oakfield sampler, we basically, in quadrants, we did composites and analyzed, processed the soil for total phosphorus, malic, water extractable, and then this is one of our interns' cat harvesting plants. We dry the plants, grind them, here's dried elderberry, and analyze those phosphorus concentrations. In terms of mycorrhizae, we gathered the roots, we made these slides, and then we dyed them, and then we looked in the microscopy at these organelles for quantifying colonization. And in terms of DNA, we submitted some of our samples to colleagues who do DNA sequencing, and then for pollinator habitat, we did transects a couple times every month of the growing season. So let's talk about data. One of the things that's worth noting is we wanted to check our assumptions. Were the inoculated plots actually look inoculated, even though we clearly inoculated them, 2,200 plants from inoculant we grew? And yes, indeed, we see, if you remember, the green is the inoculated. Think of M. mycorrhizae. The RV is uninoculated. Statistically significant, more inoculant, the y-axis is the amount of colonization in the inoculated plots. Good, that's what you would hope. However, look how high that concentration is in the uninoculated plots. That's really high, and that's because we left all the native species in place when we restored, and those plants, of course, have mycorrhizal associations. So do the buckthorn, but those don't necessarily play well with many plants. Now, this is not as overwhelming as it seems. I'm just giving you the 3 different soil metrics we talked about of total water extractable and malic phosphorus, but what is worth looking at is the y-axis are just the phosphorus concentrations. These are the dates of soil sampling. All 3 metrics, and this is just this year's data, show the exact same pattern. Statistically significant less phosphorus in the 2 restored plots compared to the control. Okay, that's good. That means that restoration is mitigating phosphorus. However, no statistical significance between the inoculated and the uninoculated. Now, you can look at the data a different way, and you can look at how does fungal diversity affect phosphorus concentrations, and we see a very interesting trend here in both the water extractable and malic, and that is when you increase fungal diversity, you decrease phosphorus concentrations. So then you think, okay, how do we apply this as practitioners? That means we want to increase fungi in the landscape, obviously the beneficial ones that partner well. And how do you do that? Plants, diversifying plants. And when you're thinking about that, you want to make sure that you're working with plants that have both specialist and generalist fungal partners. If you're not familiar with the phosphorus cycle, it's very complex. Unlike carbon and nitrogen, it doesn't have a robust air phase. And 90 to 95% of phosphorus is locked up in the soil. And so that 5 or 10% that becomes soluble and can move into the plants and be uptaken is really done through the enzyme exudates, the chemicals released from the mycorrhizae or those microbial communities. And that's something that's really important to think about, because that's when they're actually in the plants is when we can harvest them and remove them from the landscape before they go senescent and go back into the soil and the water. So this is a guide that's free on the SARE website, and it basically goes through how to grow mycorrhizae fungi. Now, when we originally did this, we gathered soil from the wild part of the Shelburne Farms forest. But the original study was done when we wrote this guide was for Digger's Mirth. You may know a wonderful farm down at the Intervale, because we were putting in mycorrhizal banks there. And that was the appropriate to use cover crops. We used red clover, alfalfa, and oats. However, that's good for an annual crop production farm, the edges of it. But I would not recommend using cover crop seeds to grow endemic mycorrhizae for a forested riparian buffer. And this is why. If you look at our DNA data, we'll just focus due to time on these last 4 bars. And what you see is this genera of fungi, glomus. And by the way, if you go into your gardener's supply store, the cat's out of the bag, you're going to see commercial mycorrhizae everywhere, and that's very problematic. It's more problematic in Central South America, where literature indicates that non-native mycorrhizae are actually facilitating invasive trees going into forests. We're not at that level up here, but it's still problematic for the reasons shared earlier. And so you see that from year 1 to year 2, this glomus is basically moving in, and the clariglomus and the paraglomus are getting smaller. And remember, we talked about that primary succession? It's happening in the fungal community, and that has implications. So that is why, 1, we're not using commercial mycorrhizae, but 2, if you're going to grow mycorrhizae, you want to grow it on the polyculture seeds that you're planting so that you can maximize pulling out those diverse fungal communities. Secondly, you might not even need to introduce mycorrhizae in forested riparian buffers if you leave native species in place. The place I think that this technology is more appropriate are over-fertilized, over-tilled edges of fields that are just completely degraded underneath to jumpstart, and that you're following them then with the perennials and grow it on those perennials. So another interesting thing to look at is fungal succession. And what you can see here from our DNA data is that from year 1 to year 2, there was definitely fungal succession and diversity going on. Another interesting thing to look at is the relationship between over 2 years, the phosphorus concentrations, because remember that earlier data I showed you was only last summer. And what you see from year 1 to year 2 is that the fungal, I'm just going to take a breath here. I'm sorry, I'm racing because we lost several minutes with the technology. What you see here is you see that the phosphorus concentrations decreased in both the malic metric and the water extractable metric. And I'm happy to break down those, what those different metrics are later. And you see a bigger decrease in the inoculated and plot. So that's very interesting to think about. Would you see this translation in the plant uptake? So let me just back up for a second. So if you follow that logic, if there's more phosphorus decreased in the inoculated plot, would that mean that you would have more phosphorus in the plants in the inoculated plot? Like, where is it going, right? Maybe you're going to see it in the above ground body. Well, in our pilot, we certainly didn't. Just breaking down, the y-axis is phosphorus concentrations. Yellow is buckthorn, by the way. Buckthorn does not take up a lot of phosphorus, in case you were wondering. But these native species, there's really no statistically significant difference, right, in the plant uptake. Now, if you look at our new study, there was for bone set. Okay, that's cool to know. There was definitely more phosphorus in the bone set. But for all the other species that we gathered and analyzed the data from, there really wasn't a big difference, okay, in this particular case. And we have a couple of ideas of why that might be. However, I will note that these calculations are very valuable because we can do back the envelope calculations of how many milligrams per kilogram of hectare of phosphorus can be removed when you harvest different plants. And you do that throughout the watershed. We're all doing that at non-point source places. Hey, that's phosphorus mitigation. We cycle it up the ecosystem. We provide food and medicine security for marginalized groups. Doesn't necessarily just need to be the Abenaki. It's definitely something to consider. So what about plant diversity? Well, it doesn't really look like in the pilot there was a difference. In fact, the first year there was more diversity, these are counts, in the inoculated, but the following 2 years there was actually more in the uninoculated. And that could be because of what I told you about the parasitism spectrum. We also have two other hypotheses that I could talk to you about later. The other look at is this year's data, the current expanded study. Again, of course, we see statistically significant more counts in the two restored plots compared to the control, but really no difference. And even with those that migrated in, no difference. And we define migration in this case as coming up from latent seabeds, coming from wild ecosystems. So let's talk about phytoremediation for a moment. We mentioned that phytoextraction is a form of phytoremediation that we're practicing. And this is where a lot of you practitioners don't get funding for harvest and maintenance, but that this could really help increase the phosphorus mitigation of our buffers. And because we work with the indigenous people, we're not doing all of it. We're doing the honorable harvest, as you probably heard from Robin Wall-Kimmer when I learned in herb school years ago, right? Only a quarter each year to share with the beyond human community. So again, I mentioned these calculations. All of these plants have different uptakes, and that's something that we are hoping to work with the Phosphorus Pays 2.0 program. Pollinator habitat, as Spencer mentioned earlier, leaving some of those hollow stems for the pollinators. And then, of course, food medicine tools, community security, right? People are marginalized in their ability to obtain their own food and medicine. This provides a wonderful opportunity because the farmer doesn't always have time to do this. And then, of course, those doing the work have wonderful opportunities to grow eco-literacy. So I just want to mention this fourth paper we'd written about the work. There's an important word here of balancing. And I bring that in because when I was originally doing research on phytoremediation, I was looking at farms that are growing willow for biomass, where they're just harvesting all of it. And if we were to do that, we would have higher phosphorus removal counts. But then we wouldn't have the biodiversity and the indigenous land access. So it's always balancing these objectives that we have to think about in terms of how much we're harvesting and the other aims. This is just a quick little thing that I think people might find interesting who work with plant palettes. We wanted to ground truth our plant palette. So the dark green represents what you would see in published palettes of what would be blooming. The lighter green shows you what actually was blooming. And it was really cool to see that there were actually more blooms in June and August than the palette indicated, but less, right, in May and September. And when you break down the light green into those planted and migrated, it's fascinating to note that in June, in July, and September and October, the migrant species were ones holding it down for the pollinator community. And again, that's just so important, right, when we're restoring ecosystems to do as little disturbance as possible because the natural intelligence, right, of those organisms and relationships are already going on. And so this is just a snapshot of some of the flowering, numbers of flowering each time we went out. We were hoping for, more than 4 each month, or at least 4. So hopefully when the ailanthus and witch hazel are blooming, which hopefully will be this year, those numbers will increase a bit. So our findings, endemic mycorrhizae might not necessarily be helpful in riparian forested buffers. Restoring buffers through the non-chemical removal of and again, only for buckthorn and honeysuckle, this is not a strategy for Japanese knotweed, can facilitate phosphorus mitigation following that 3 times in 2 season approach. Designing, installing, maintaining this native multi-synusial, right, tree, shrub, herbaceous ground cover can increase pollinator habitat, facilitate succession, and co-designing a palette with the original peoples can offer a model for reciprocally involving marginalized groups. That's the work of our generation. Now, I just want to briefly end with looking at some questions because everything we think we might know, we actually is a doorway to what we don't know. So, endemic mycorrhizae may not be helpful in this type of riparian forested buffer. Is that because we saw that there were already endemic mycorrhizae present? And so, or is it because we grew them on cover crop seeds and we didn't grow them on riparian polycultures? In restoring the buffers through non-chemical removal, we saw significant decreases in phosphorus in those restored plots. Now, what if we just did the strip back and we didn't remove the stumps? Because I don't know if I mentioned that the wonderful people that helped us, we had hundreds of people come out over the year and the pilot, those 2 different projects help, many work parties, and they were digging up the stumps away from the waterway. So we're wondering, were those phosphorus decreases because of the stumps? But if we just did the strip back, would we still have those phosphorus decreases? And what other species do we want to get phosphorus metrics on? I recently wrote the agroforestry working group on this, and I don't know if people probably know Jesse Markinson of Yellowbud, you know, the big bitternut revolution we're going on. We're definitely going to get data on that bitternut hickory, and if you other species you think you want phosphorus metrics on, let's learn it, and we can get the data and analyze it and have that to add to the phosphorus removal matrix. Designing, installing, maintaining native synusial palette can increase pollinator habitat, but we never got metrics on pollinators. We are going to have interns start that this summer, but I just want to note that this study is ending December 26th. And after that, you won't have to follow foot hygiene that we do now, going between the inoculated and uninoculated plots. We're writing a manual for the farm, and we're opening it up to everyone to, regardless, you have to first ask Shelburne Farms permission to continue research there and giving it to the community. There's a lot of wonderful opportunities, and particularly in pollinators, to look at in those plots. And finally, co-designing this palette with original peoples, offering a model for reciprocally involving marginalized groups. What would this look like doing across the lake with the Mohawk and all their non-point source places and their plants that are edible, medicinal, utilitarian to them? What about in Canada, the Odanak, right? This is just reclaiming the commons, right? It's like how we used to live before the colonial era, what we're trying to get back to. So I just want to name that there is a huge amount of people that have been incredibly important and supportive to this project, and organizations and funders. We're super grateful to them, and we hope that we honor all their efforts. And with that, if there's time, I'd like to offer time for questions. I'm sorry for the technological kerfuffle. And I just want to name that we're having a site tour July 23rd. It's free. If you ever want your group, your school, your organization to come do a site tour or help us remove buckthorn around the plots, we're happy to host you. So please feel free to reach me. And this is an INOT project, one of our eco-literacy tools that our interns are working on improving and including pollinators on. You may know Desiree, Vermont Eco Studies has the PIP program in iNaturalist, so we're trying to now link the plants with specific pollinators. Thank you so much. Any questions?
Thank you, Jess, for that amazing presentation. I was curious about to what extent the research was advised upon by the indigenous communities and what that involvement looked like.
Yeah, thank you so much. It's been a huge part of the project since the start. So the pilot project started right before COVID, and then COVID hit, and I was like, we're not stopping. So we just did it through MycoEvolve. We couldn't do it through UVM. And so Microbial, the organization that I facilitate, stay with my interspecies community, we wrote a small grassroots grant with New England Grassroots Environmental Fund, and we worked with the Abenaki. We paid them because, we're not going to ask them to do it for free. And they did a rematuration ceremony at the base of the site to the lake. One of my elder friends had kind of a dream of a vision of facilitating rematuration and asked permission, and we did a beautiful ceremony. A lot of community came out. And since then, we've worked with various partners. Now, I'm sure you're aware there's a lot of politics going on. I'm not going to get into it, the Abenaki. And so I first worked with like the chiefs, and then I worked with like different tribal members, and eventually I just ended up working with the women. It was much easier, and they're the medicine keepers, and it just flowed. And so I ended up working, at least for the expanded pilot, the expanded project, where we tripled the plots, and our palette was 100% edible medicinal utilitarian with the Abenaki. I worked with Carol McGranahan in particular. And now we have the next generation of Abenaki because we wrote them into the grant. I should note that thank you to Sea Grant, we were able to triple the research. And so the next generation is actually going to do a little different thing this summer than she did with her generation, but she wanted to hand it off to them, this last generation. And so Carol went out there. I don't know if I had a slide, probably not, so I was trying to make it short, but we had like Abenaki education programs in the summer. So Carol would take Abenaki out, she'd show them the sweetgrass, the tobacco, we'd harvest it, we'd make elderberry syrup, she'd do a whole plant slide and talk about the colonial plants and the native Abenaki plants and how, all those different things. And this year's generation, they want to make a bunch of medicine and bring it to all the elders. I will note that a lot of the Abenaki don't want to set foot on Shelburne Farms. And most respect to them, the Shelburne Farms community, and all the wonderful work they've done. And there's just colonial politics and a lot of trauma there. So it's been humbling, complex, but I feel like it's kind of the least we could do to try to reconcile, this colonial history that we've inherited. So thank you for your question.
Thank you so much.
All right, well, I don't want to take any more of your time, and thank you so much. And I'm happy to connect with you more. I'll be around for a little while. Thank you.